Good afternoon and welcome to another PSDI webinar, BioSimDR, a collection of data tools and infrastructure for biomolecular simulation. My name is Dr. Mansoor Nelikal and I'm an enterprise fellow at Physical Sciences Data Infrastructure, PSDI, and I'll be chairing today's session. If you're new to PSDI, it's a UK-wide community and platform that makes physical sciences research data accessible and reproducible by providing various tools, guidances, and training to help researchers to collaborate and accelerate discoveries. Now, let me introduce our today's speaker. It's my pleasure to introduce Dr. Jas Kalayan. Jas is a scientific software engineer at STFC who works on making research workflows more reproducible and improving how data is shared, particularly in molecular simulations. She has a background in molecular modeling, including developing machine learning methods and approaches to better understand complex systems. Her work has supported studies in areas such as protein-ligand interactions, hydration, and free energy calculations. Overall, her focus is on making computational research more transparent, reliable, and easier to reproduce. Thank you very much for joining us today. Over to you.
Thank you so much, Mansoor, and hello, everyone. So yes, as Mansoor said, I'll be talking about biomolecular simulation data resources, so BioSimDR for short. And so in this talk, I'm going to firstly introduce what biomolecular simulators do and the challenges that's faced with how they handle data. So that will be sort of a brief introduction. And then sort of the current data infrastructure that's available for biomolecular simulators. Again, it'll be quite brief, but that's kind of on purpose because then the main bulk of this talk will be how we're addressing this with BioSim Data Resources. So an overview of the tools and services that we're providing and when we hope they'll be available. Okay, so firstly, biomolecular simulators, what do they do? So just as a quick background, they take static 3D structures resolved from experiment of biomolecular simulation. I say experiment, but now we also have predictive structures that we can take. And then what we do is we apply the equations of motions to explore the dynamics of these complex and large systems. And so what you end up with is a trajectory of the motions of the molecules in your system. And the trajectories are analyzed to understand biological questions such as those listed here, like drug discovery, protein function, protein-protein interactions, and proteins and membranes. So this is kind of what we do as simulators and sort of the interesting science that we try to capture from the dynamics of these molecules. But what else can we do with the simulations that we produce? So other things that you can do with simulations is you can train machine learning models to predict molecular conformations and interactions. You can apply ML models to larger, more complex systems, potentially running them quicker or even more accurately. You can also use simulations to generate synthetic data and use that as a ground truth to improve interpretation of images that are usually produced from experiments. So here's an example of a synthetic electron microscopy micrograph. So something like this would usually be produced experimentally, but we can create, a synthetic version of it and then test the algorithms that are used to analyze these sorts of images. You can also use simulations to help validate and understand experimental data at atomistic levels. You can also use simulations to develop new methods to analyze simulations, such as looking at new ways to look at interactions between molecules, maybe a new method for free energy calculations. You can then also take a simulation protocol that has been published or is of interest to you and then apply that to new systems of interest. The problem with that last point, though, is the simulation protocols that are produced are quite bare compared to what it is that we actually do. So simulations involved many different steps to produce them, and the workflows involve many considerations in each of those steps. And also we have many tools that we can use in each of these steps. So for example, you will build your molecular system, so what's actually going into it, and you have these considerations of what structures you're using to begin with. What are the orientations of these molecules with respect to each other? What kind of waters are we introducing? Are we preserving those that are found in experiment or not? Are we going to use predictive structures? Are we going to use experimental structures? All of these considerations are just for, starting off with your molecular system. And then you need to consider the physics of how we represent the interactions between the atoms in the simulation. And then you need to consider relaxing that system, minimizing the energy between your atoms. And if this fails, then you then often need to go back and rebuild your system. And so really what I'm trying to illustrate here is that it's not just as simple as running something once and it working. It's often an iterative process of trying to take a system of interest and actually getting the dynamics to be performed. And once you have performed your simulation and you've considered all of these sort of settings that you need to include for performing your simulation, then you will go on to analyzing those dynamics, look at various things like hydrogen bonds. You might want to visualize certain interactions. And like I said, there's so many tools that are open source and available for us to use. And that is one benefit of this field, is that we have so many tools that we can use to produce our simulations. But the problem is when it comes to sharing our data, you can be a bit lost in how something was performed. And so reproducing this is often quite difficult. So here's an example of what describing a simulation protocol looks like in a scientific paper. So you often have what was done and what was used, but then there's often quite a lot of missing information. And that's of no fault to the person publishing this. Often there's limited space in what you can include in describing your workflows, because the main point of this is the interesting science. And often you don't want to, because it's too convoluted to include all the variables that you want to include in a scientific article. There's also no standardized way to represent these protocols. And so if you were to describe this fully, it might not be machine readable because it's not consistent with how others would describe this protocol as well. So all in all, when we produce this data and publish our results, can we reproduce the interesting science that we have published? And usually the case is no. Even this is something that I published. Would I be able to reproduce the simulations that performed in this paper just from looking at what the protocol was from the paper? Probably not. I wouldn't be able to reproduce it. Another issue we have is sharing the data that we have. So there are places to share, such as large general purpose repositories or even smaller field-specific databases. But when you share your data, it's often difficult to find simulations because they're mixed in with many other data sets from many other fields. There's also a lack of tooling to capture the metadata that is used to produce these simulations and also outputted from the simulation. And so you can't really capture that provenance in domain-specific ways and share that with these sort of general data repositories. So it's left to the researchers to decide what to contribute. And so there's no consistency in how and what we share. Also, some of these services are quite strict on how much data you can upload. So in our case, molecular simulations can be quite large. They can be many gigabytes to terabytes of data per simulation. And so these smaller databases and even these larger ones, it's not feasible to be able to share this data more widely, even if you want to. And it's ironic because the other areas of biology are really good at sharing data, and we often use these data sources to start our simulations. And it's only really fair that we also try to share our data consistently and allow other fields to use our data. And so what does this sort of current landscape look like for data infrastructure for the BioSim communities? And so in the UK, the research communities, funders and support looks something like this. So we have UKRI that provides the funding via EPSRC. We have two main research communities on the right. So CCP BioSim, which focuses on new scientific codes and focuses on building the community. And then there's HEC BioSim, which focuses on high-end computing and HPC engineering. So both of these communities run biomolecular simulations. They're approximately representing 1,000 researchers in the UK across 63 institutions. And the funding is usually provided via COSEC for CCP BioSim anyway, which is focused on providing software as an infrastructure to various communities, including BioSim. And so there hasn't really been sort of data infrastructure support for these communities until PSDI came along. So PSDI is providing ways to, link existing and also provide new physical sciences data infrastructure. And through the various partner projects involved, one of them is the Biomolecular Simulation Data Resources. That's headed by James, and this is what I work on. So what I'm going to go through in this talk is sort of the 5 main areas of how we are supporting data infrastructure for BioSim. And I'll go through each of these and how they are connected. So the first one will be data recorders. So as I mentioned previously, we have these complex workflows where we're using many different kinds of tools. And ideally, we want to be able to capture exactly how we do something. Everything we do is pretty much computational, so there should be a way to capture this. But because our landscape is so varied and the software and hardware that we use is so varied, it's very difficult to capture this. So we want to know, where simulations are performed. Are they performed on HPCs, clouds? What environments are those in? Are they in virtual environments, in a Docker container? What software, hardware is used to run the code, and then the software programs that are actually used to perform, set up, and analyze the simulations. We want to try and capture all of this to make our data truly reproducible. So the first way in which we are doing this is to use a tool called IEDA to capture the data provenance of our simulations. So IEDA is a Python infrastructure, and it helps to track complex workflows used in computational science. It's developed by Giovanni Pizzi's group at PSI in Switzerland. And the idea really is we use IEDA to wrap simulation commands into the workflow engine. So we've taken the 2 most popular simulation engines used in BioSim, so that's GROMACS and Amber, and that represents about 90% of our users. And we have wrapped the commands that are run in these programs into IEDA. And what this basically allows you to do is it allows you to sort of run your commands as you would have done when you're performing your simulation. But in the background, IEDA is able to actually extract important information about how a simulation or how sort of commands are run in the background without a user having to really worry about it. So what IEDA does is it will create a database and it will run a daemon. And every time you run a command via one of our plugins, it will say, okay, I can see that there's a command coming in. I'm going to check and see what are the inputs that are required for this command. Have they been provided to me? And if so, great. I'm going to now run this command in the simulation engine that the user wanted me to. Then I will check and wait until this command has finished running. Once that's happened, I'll verify the expected outputs that were produced from this command. And then what you can do within IEDA is you can have custom extractors where you can extract useful metadata from the files that have been produced. You can store that in the database, and then IEDA will return the outputs of that command back to the user from where they submitted. So all of this is happening in the background. The user is kind of none the wiser, but you're able to extract this useful information about what was used, what was produced, what compute was used. All of this information can be extracted without the user having to worry about it. And then you can sort of iteratively carry on running your simulation workflow using IEDA. And what's nice about it is you can choose what commands you want to track. You can have a look and see what is being tracked. You can delete things if you want to. And once you're happy with your workflow, you can bundle that up into a single file. and then share that wherever you want to. And this is what it looks sort of like in practice of what a command looks like when you run it through one of our plugins. So natively, this command that is run for performing a simulation in GROMACS looks identical. The only difference is we've included this underscore, and that is how we've designed it to make it look like the native commands. So we hope that it looks familiar to users, so it's a low barrier to adopt. And then the complexity, like I said, is abstracted from the user. Another thing that you can do is you can include it into an API. So if you run your simulations programmatically, you can include this in a Python script. So you can have automated provenance capture, and it's Python based, so it's sort of easy to use by scientists. And so what's actually captured by AIDA? So what you will see is a graph representation of the data that was captured in the background. And what you have is a command that was run, and then you have information about what that command was, what software was used to run that command, where that command was run from like a local computer, and the inputs that were expected for that command. And then from that, you see outputs that are produced. And if you build the picture of your workflow, you will see that you can produce these large provenance graphs of how your various inputs and outputs are connected to each other to form your fully connected workflow. So here are some examples of actually using our provenance tools in practice. So Kin, whilst he was a graduate student with us, ran simulations of GPCR proteins. So these are proteins embedded into a membrane, and he was looking at the interactions of lipids on these 15 proteins out of 800. And his workflow involved taking a structure, removing regions of low confidence, orientating that protein correctly so that we can build a membrane around it. He also used tools outside of GROMACS. So he used something called Martinized 2 to sort of coarse grain the simulation. And then he also used a custom script to embed the protein into a membrane. So each of these steps we were able to sort of modify so that we can capture them via AIDA and form an unbroken provenance workflow. And so his data is shared in our data collections, which I'll talk more about later in this talk. We also have a tutorial describing exactly how this workflow was produced on our GitHub workshops. And then also the scientific article highlighting using our provenance tools to capture the workflow for this particular simulation. And he's ran this for 15 GPCRs, but because we know exactly how he performed the simulation, you could apply this to all the others that are out there and extend this study if you wanted to. Another example was done by Joel. So he is not a biosimulator by trade. He wanted to run simulations, though, to produce the synthetic micrographs that I touched on earlier. And he wanted to run a simulation where he's sampling this hinge motion of this protein. And so you can run a native or a standard simulation, but you wouldn't see or capture a lot of this flexibility in this hinge region. So he ran some enhanced sampling simulations. Again, we were able to capture these enhanced sampling simulations using our provenance tools. So although we're using a plugin called Plumed to run these simulations, we can still capture the provenance of it, even though it's not natively a GROMACS tool. So although Joel hadn't ran simulations before, it was a good test to see how easy are these tools to use for someone who's a non-expert. And I think one of the things is if you can get your software installed correctly, so you need to install AIDA and our plugins, and once you have that set up, you can then pretty easily run simulations and track the provenance with our tools. Okay, so there we have our sort of solution to recording our complex simulation workflows. Next is describing the metadata that is used to produce these simulations. So we want a consistent way to describe our simulations regardless of what tools we're using to perform them. So we use a schema to consistently describe and present simulation metadata. And in developing this schema, where we can, we will use existing ontologies to define the terms that we use in our schema. The problem is that for biomolecular simulation, we don't really have any ontologies out there already. So one cool thing is the MolSim ontology developed by a group in the University of Düsseldorf are developing an ontology for molecular simulation. And this ontology is also going to be used by Molecular Dynamics Database, which is a European initiative to share molecular dynamic simulations with the wider scientific community. So this is something hopefully we can use in both of our projects. So here's an example of what the biosimulation metadata schema looks like that I have been developing to represent biomolecular simulation data. So it uses a tool called LinkML, and that's linked data modeling language. And it allows you to create a schema using YAML. And YAML is quite a nice, intuitive language to use. And it allows non-experts to also contribute to the schema. So I wanted to use a tool that meant that anyone in the BioSim community can add to this schema if they wanted to. And it allows users to define, validate, and share data structures in a machine-readable way. So when you have created your schema in YAML, you can then also output your schema into other various formats that are more machine-readable, such as JSON-LD, GraphQL, Shackle, etc. So this kind of looks complicated, and I'm not going to go through it in detail, but one thing I did want to show is sort of what we're trying to include in this schema. And so an example of a simulation setting, such as like a cutoff distance, we want to be able to describe what that term is, define, what it is, so what kind of quantity it is, what kind of units you should expect from it. And then importantly, within the schema, we have also included mappings to various MD engines. So we want to describe something consistently, and then we want to know how it was described in an MD engine. So in GROMACS, this particular cutoff is described using this key with this unit, and how it's described in AMBER is given here as well. So what we can do with this schema is we can extend this to other simulation engines such as LAMMPS and NAMD, OpenMM, whatever we want to map it to. We can then add these various mapping terms and ensure that our schema can be representative of the various simulation software that we use in this field. So we also try to map to ontology URIs where we can. So for example, the units will define the meanings of them using existing ontologies, as I mentioned before. So this leads nicely onto actually extracting the data from simulation output. So I mentioned just now about mapping to MD software. And so what we can do with these extractors is we can use the schema to tell us what to extract from our simulation outputs. So for example, we have these input files for AMBER and GROMACS, and they describe terminology in different ways. So anything that's highlighted in the same color is representative of the same quantity, but described using a different term and also in some cases different units. And what we will do with our extractors is to extract this information and then use the schema to define it in the same way using the same units. So here's an example of some extracted metadata that's aligned against the schema that I mentioned. So we have our own Python packages to extract data, and then within those packages, we use tools like MD analysis and RDKit to extract further information about the molecules in the system. So, for example, we can use the log files from the simulations produced to output observables such as energies and pressures and temperatures, and then provide some standardized units for each of them. Then we can also use MD analysis and RDKit to extract molecular information such as, how many molecules of a certain type there are, how large that molecule is, information about, for example, what its SMILES representation and INCHI key is, so that this can be linked to other databases as well. Okay, so that's the extraction of data and presented in a formalized and consistent way across the various tools that we're using. The next is to share that data via an API and GUI. So what our plan is to allow users to deposit simulations via a graphical user interface directly embedded into the CCP BioSim website. So the idea is that users can just upload files that they've produced from their simulation, and it's usually a topology file which describes the composition of your system, so what atoms are in there, how they're connected, and then also the trajectory of the dynamics. So these two files users will most definitely have, and provided that you can read them using MD analysis, and MD analysis provides many different readers for various formats of simulation files, so it's quite extensive. And then if you want to, you can also provide your provenance file via AIDA as well. So all a user would need to do is just click extract, and it will extract the metadata according to our schema. And then what do they do with this metadata? They can either download it, use it, include it with their simulation data and share it wherever they want to. Another option is that they can submit it directly to BioSimDB, which is the repository that we're using within PSDI. I also wanted to add that you can also submit data via an API to BioSimDB or to any of the data collections, which I'll talk about later, that are provided by PSDI. And this API has been developed by Elliot Jacob in Alan's group. So onto the data repository. So that's BioSimDB that I touched upon. So we also want to include a place for people to upload their data if they wish. And so PSDI offers the data collections, which has been built by Andy in Gemma's group, which uses Invenio RDM. So if you've used Zenodo before, it's the same backend as that. And the data collections is a collection of community repositories, and one of these repositories is BioSimDB, so it's specific to biosimulation data. So the deposited data is available to anyone, and depending on the license that you attach to your data, users can use it how they wish. Data deposition is open to UK-based academic institutions. There's no hard limit on the size of the data, although, if you want to upload anything over 100 gigabytes, then maybe talk to us first. We have currently 2 petabytes of storage available, which can be expanded. So if we have users that are actively using BioSimDB, then we could expand that and allow for others to use this resource as well. So at the moment, it's in its early stages, and we're keen to have people test it and upload their data. So another thing that we need to consider with BioSimDB is the community-led data curation. So we want to ensure that the data that is submitted is of high quality and consistent. So here is a sort of workflow of how this could potentially work. So users would upload simulations to BioSimDB. via the GUI or via the API or via data collections itself. And the idea is users, if they used our GUI, then they can extract that metadata automatically. And the only thing they'd need to fill in is just information about the authors, any licenses, information that, we can't really extract automatically. And then once you submit your record for review, BioSim data curators can check the fields are filled in correctly. So we want this to become an automated process. So we can then use, for example, the extractors just to double check that the fields that are specific to the community are filled in correctly. And then the fourth part is probably where you wouldn't want to automate it, is to check for scientific relevance. So is the data that's being submitted useful to the community and beyond? And then once a reviewer is happy with that, the record can be published to BioSimDB. So this curation process is something that we still need to think about a bit more. And with the help we've had from Karen and Elizabeth to sort of understand what is involved in curation, it's not like a simple thing to do, but if we can automate processes and ensure things like we're using standardized units, standardized descriptions of our metadata fields, then this should be a more straightforward process. Okay, so that's kind of all of the 5 components of the data resources that I wanted to talk about. So we have the schema, the extraction tools that extract metadata according to the schema, the provenance tools that are used to capture the steps that we use to perform our simulations. We also slightly touched upon training and resources documentation, training resources and documentation, which will be provided for all the tools that we produce. So at the moment we have tutorials on how to use our AIDA plugins that are available on Jupyter Lab and Docker containers. And then also a place to deposit data. So we have various ways in which you can deposit using an API, a GUI, or you can deposit the data in any repository that you see fit. So in terms of availability and the status of the resources, so the schema and extraction tools will hopefully be released this summer, and we will make these available openly to use and contribute to on GitHub with obviously the relevant documentation. The data provenance tools are available to use and contribute to, and we will also update these with the new extractors that conform to the schema as well. As I said, training, resources and documentation will be included in all tools released and will be extended as and when required. If we have any new features, we will update documentation accordingly. And data deposition. So at the moment, BioSimDB is in the beta phase, and what I mean by that is anybody who's from the UK can upload data, but they'll have to manually fill in the BioSim specific fields. The API is also available, which isn't specific to BioSimDB, so anyone who wants to upload to any of the data collections in PSDI can use this API that we've developed. We will update the BioSimDB with the new schema later this summer, and then also integrate the GUI to upload and extract metadata later this year. And then also data curation is, to be determined of how we properly do this. So the final slide is what will all of this enable? So providing this data infrastructure for the BioSim community. Hopefully it will provide all of these things that I'm going to talk about now. So we want to provide tools to record the complex workflows that are performed in this field, and then be able to then share these blueprints with others. And so allow for the full provenance of simulations to be more readily available. So if somebody is leaving a lab, for example, that data and knowledge isn't just lost with them. We still have it stored somewhere in a consistent way. We also have provided a way for simulation data to be stored in the UK And this will allow for tools to be developed that are compatible with the next and current generation of HPC. So, what do I mean by this? Hopefully, we can upload data directly from HPCs onto BioSimDB, for example. We also are looking to reduce duplicated effort and improve statistical aggregation. So there may be various sources of simulations that have been submitted of the same system, but they might be representing different dynamics, and you can combine those to do a larger study of the dynamics and analysis of these systems. We're also going for something where we are more greener for the environment. So instead of regenerating the data, we can just reuse what we already have. And as these systems can be quite large and take a lot of compute to perform, it is actually greener to reuse than to reproduce them. And so, lastly, we want to provide a data source for new and innovative fields. So, artificial intelligence and machine learning. So, most of us will know of or have used AlphaFold to predict structures of proteins, but these are static structures. What is the future for dynamics and being able to predict this? We provide a standard data source to test and verify new methods and technologies. And with this, because we can provide standardized ways of describing things and providing that, hopefully knowledge will bleed through the community. So we don't have to keep reinventing the wheel when somebody new joins a group. We've got these protocols ready to use and extend by new members of the community or other people who are interested in contributing to the community. Okay, so with that, I'd like to thank you all for listening and thank everybody again who's worked and helped out in this project.
Thank you, Jas. It's an interesting presentation. Now let's open the floor for questions. Does AIDA works with GROMACS patched with other softwares like Plumet?
Yeah, so with Joel's study, he wanted to use enhanced sampling, so we made sure that it works with Plumed. So it does work with Plumed.
Mm-hmm.
If you have another plugin that you are using with GROMACS or Amber, whatever, then we'd have to extend the provenance tool so that it does work with it. So it's kind of as and when basis. If that's a feature that somebody would like, where we want a plugin to work, we want a simulation engine to work with a plugin.
Mm-hmm.
That can happen. So yes, it can happen, but the thing is that when you introduce a new external tool, then you need to understand how it works and make sure we definitely can track the expected inputs and outputs for that new plugin. So yeah, it is possible to do.
Could you please elaborate on the community data collection? So are there existing data? What kind of data is available for download, etc.?
Yes, so at the moment in the BioSimDB, we have sort of migrated data from a small database called
Mm-hmm.
SlimMD. So SlimMD is a database of simulations that were produced during COVID for used for like educational purposes. So at the moment, you can find that in BioSimDB and download and use those for free. And what we're hoping to do with BioSimDB is to have sort of not like large curated data sets, but more, individuals who are interested in sharing their data, sort of ad hoc can share this in BioSimDB. So it's more, an accessible way to share
Mm-hmm.
data is what we're hoping BioSimDB will be.
So thank you once again, Jas, for giving us the wonderful talk. So the PSDI resources are live now. If you're new to PSDI, if you go to our website, you can see all the data sources, tools, etc., especially the ones which Jas mentioned in the talk. The Amber and GROMACS plugins can be found under the tool session, and the data sources are available to access. So please do take a look. We are having a few more events lined up. We are introducing a new webinar series. This is to introduce the outputs and resources from our funding call 2025. So it's a series of webinars which introduces new tools and resources produced by the new partners of PSDI. So the session 1 is starting on 30th April 2026. So please do visit our events page. You can register there. And you can also follow us on our social media for new updates and events. All the webinars, previous webinars are available on our YouTube channel. You can see those, watch it. Thank you again. Thank you all for attending. These are our social media pages. Please do follow us and see you all in next webinar.